Hello, students. Thank you for tuning into this lecture. This particular part of the lecture is on Chapter 8, but it is specifically dealing with genetic transfer and recombination. Typically, when we say recombination, we are referring to the exchange of genes between two DNA molecules to form new combinations of genes on a chromosome. So recombine, the term is coming from combining genes from two different sources. So again, recombination is combining genes from two different sources. And then there's another term up here talking about gene transfer. And gene transfer would be passing the genes from one cell to another. Now, this can happen in two different ways. First, we have vertical gene transfer. Vertical gene transfer is having genes transferred from one parent to the offspring, from a parent to the offspring. So that would look something like this, where you'd have the parent cell, and it gets passed on. The genes that are in there get passed on to the generation following it. So one generation is passing on the genes to the generation after it. That is vertical transfer. We do that, plants do that as well. And of course, bacteria. Now, bacteria do not only pass their genes to the next generation, and that would be vertical gene transfer, passing it to the next generation, but there's also horizontal gene transfer, which they are able to accomplish. And horizontal gene transfer would be between bacteria within the same generation. So that's genes being passed from this cell to this cell, and they are within the same generation. So what we call this cell would be the donor, and then the cell that's receiving the genes in this horizontal gene transfer would be considered the recipient. Once transferred, part of the donor's DNA will be incorporated into the recipient's DNA. So that is horizontal gene transfer. The part that is not incorporated into the recipient's DNA just gets degraded. And then the recipient will take the donor DNA and integrate it within its chromosome. And this is called a recombinant. When the cell becomes a recombinant, they can receive a lot of great things. So firstly, it can lead to something that's beneficial. So the donor cell could possibly send over genes that allow the recipient cell to have, let's say, some sort of antibiotic resistance, for example. Or it might give it some new virulent factor. Maybe it can acquire a capsule. So recombination can lead to some really nifty things, so to speak, for the recipient cell. The mutation is more likely to be beneficial, so we can get something great, like I mentioned, and it's less likely to disrupt genes. And again, the fact that it's a recombinant, it will provide new combinations of genes. New genes means new characteristics, so this could be great for the cell. Now, there are different ways in which genetic recombination can take place, and in eukaryotic cells, there is one way in which genetic recombination takes place, and we call this crossing over. So crossing over occurs when two chromosomes will join, and then certain segments of those chromosomes, the parts that join, the parts that are touching, segments of those will exchange DNA. So this takes place in meiosis 1, particularly in prophase 1, where we have tetrads being formed. Tetrads are when homologous pairs, one paternal chromosome and a maternal chromosome, will come together and they'll stick, just like this. And what happens is that some of the genetic material, we're going to call the blue one, the paternal, will rub off, will be exchanged with the mother's, so the paternal chromosome, I should say. And so here we have it. And now, when these chromosomes separate, when the sister chromatids separate at the very end of meiosis 2, what we'll have is one that looks like this, one that looks completely blue with a little bit of the maternal chromosome brushed off on it. Then we will have another one that looks like this, and then another like that. Okay, so these two chromosomes came from the paternal chromosome, and these two are resulting from the maternal chromosome. So this happens when crossing over takes place, okay, in eukaryotic cells in the formation of gametes. In bacteria, though, we can have genetic recombination. They have genetic recombination happening in different ways, which we will discuss. Okay, so in bacteria, we can have genetic recombination taking place through the process of transformation. So in transformation, genes are transferred from one bacterium to another, and this takes place in just like naked solution. We saw this process demonstrated many, many years ago, and it wasn't really understood during that time, but it was the scientist Frederick Griffith who demonstrated transformation in bacteria. So let's go through what Griffith did to demonstrate this. So first of all, he had some encapsulated bacteria, and remember that when bacteria have the capsules, this allows them to be more virulent. They are able to evade host defenses, okay, by evading phagocytosis particularly. So when he injected living encapsulated bacteria into a mouse, the mouse died, which is expected. Next, he took living non-encapsulated bacteria and he injected it to the mouse, and the mouse remained healthy. This strain did not have the virulence. It was avirulent because it didn't contain the capsule. Now, he then heat killed the encapsulated bacteria, and he injected it into the mouse, and of course the mouse remained healthy. He heat killed that bacteria. They weren't alive, so they were not able to cause disease in the mouse. Now, this is when things started getting a little, interesting. He took non-encapsulated bacteria. So these non-encapsulated bacteria should not cause disease. These are avirulent. They were living. And he also placed the heat-killed encapsulated bacteria, and he injected it to the mouse. Now remember, separately, the non-encapsulated bacteria did not cause disease, nor did the heat-killed bacteria cause disease. Here's the non-encapsulated, the mouse remained healthy. Here is the heat-killed encapsulated, and this mouse remained healthy. But when he put the two of them together, the mouse died. Now, Griffith, he isolated colonies from the mice in each of these four situations. In the very first situation, he found that the colonies were encapsulated. So he got encapsulated colonies from the dead mouse, which was expected because he injected the mouse with living encapsulated bacteria, and he retrieved from the mouse, encapsulated bacteria. Makes complete sense. Next, in the second situation, he took the colonies out of the healthy mouse and found some non-encapsulated bacteria. Well, this makes sense too because the non-encapsulated bacteria were avirulent. So we should have the mouse remain healthy, and we should be able to get from it be able to retrieve from the mouse some non-encapsulated bacteria. Now, they only retrieved a few colonies of it, and this is because the other ones were destroyed by phagocytes. So, third situation, no colonies were isolated from the mouse. Why? Because these were heat killed, they were dead, and so they did not grow within the mouse. No colonies were isolated. Now, let's move to the fourth situation. In this situation, remember, It was non-encapsulated living bacteria that these mice were injected with. So again, it should have been, we would expect this, where the mice would remain healthy. Also, it's heat killed, so again, we would expect for the mouse to remain healthy. But instead, the mouse died. So when the colonies were isolated from this dead mouse, what they found were encapsulated bacteria. Interesting, because the encapsulated bacteria were heat killed. So this is when this really provided evidence that somehow those non-encapsulated bacteria were somehow picking up the genes from the encapsulated bacteria, and they were becoming encapsulated. So again, somehow the living non-encapsulated bacteria were transformed by the dead encapsulated bacteria. And so these living bacteria acquired the ability to form a capsule and cause disease. And then, of course, later on, it wasn't until much later that other experiments showed that the DNA was the transforming factor in this. Well, really the basis of this is the fact that in nature, some bacteria, perhaps after death or even if the cell breaks apart, the bacteria will release the DNA into the environment, and then other bacteria, the living bacteria, will encounter the DNA, and they will take up pieces of the DNA and integrate them into their own chromosome. That's exactly what happened here in this transformation experiment. The living non-encapsulated bacteria encountered the DNA of the encapsulated bacteria, the heat-killed encapsulated bacteria, and they incorporated that virulent gene into their own genome, and then they began making capsules. So this particular slide just reiterates what I mentioned about Griffith's experiment. So what we have here is the fact that some bacteria in nature, maybe like after death and cell lysis, what would happen is they would release their DNA into the environment. So this is the DNA that's released into the environment. Now notice the genes on here, they have been illustrated using capital letters, okay? Now, other bacteria can encounter the DNA. So other bacteria, meaning this one that I'm circling in green, or this recipient cell, it would encounter the DNA. And depending on the species and growth conditions, these can take up the DNA fragments and eventually integrate them within their own chromosomes. So here you can see that the DNA, look, here's the capital letters, from the environment has been taken into the cell. And they are able to integrate that DNA that they took into the cell into their own chromosomes by recombination. Just so you know what the way that this happens, and this is a little more than what you actually need to know, there's a protein that's called REC for recombinant, REC-A. This protein binds to the cell's DNA and then to donor DNA, and it causes the exchange of strands. So it's a protein that's allowing this to happen. Now this cell that now has this recombination, because you can see here all of the lowercase letter genes, and then here is the capital letter that's integrated within its chromosome. This is considered a hybrid, or the name that we're giving it right now would be a recombinant cell. And then all of the descendants from this cell will be just like it. They'll be identical. Okay, so we just covered genetic transformation and recombination as the mechanism for that. And now there is another mechanism by which genetic material can be transferred from one bacterium to another. And this mechanism is called conjugation. Conjugation is concerned with a plasmid, which is a circular piece of DNA. So anytime we're talking conjugation, we are talking a plasmid. And this circular piece of DNA is separate from the chromosome. Now, there are a few reasons why conjugation is different from transformation. First of all, one reason, I'll just write it up in the corner here. The first reason that it's different is that it requires cell-to-cell contact. That's number one. And then the second reason why conjugation is different from transformation is because the conjugating cells have to be of opposite mating type. So I'm going to write that up here. Okay. So in other words, if the donor cell has a plasmid for the F factor, so the F factor is a fertility plasmid. Okay. So if it has the F factor, then the recipient must be F negative. So here, this is the example here. We have the F plus in the donor cell. Therefore, the opposite mating type would be F negative. So the recipient should not have that particular plasmid. So that's one of the requirements for conjugation, for something to be considered conjugation. Now, just to add to this, just to make sure you know what the F plasmid is. The F plasmid, there's in some gram-negative bacteria, the plasmid carries genes that code for the synthesis of a sex pili. And the sex pili, I'm going to circle it here and let's put it in like a blue so you'll know what I'm referring to right here. This is the sex pili. And that sex pili is a projection from the donor cell surface that contacts the recipient cell, and it brings the two cells into contact with each other. Remember, conjugation requires cell-to-cell contact. In gram-positive cells, they get like a sticky surface molecule, and it causes the cells to stick to each other and come into direct contact with each other. The very first plasmid that was observed in bacteria was in E. coli, and it was this F factor, the fertility factor. So the donor is carrying the F factor, so we call it F plus, and it transfers that plasmid to the recipient, which was F minus. Okay, so it did not have that. So you can see here in the second illustration that the plasmid is being sent over into the F minus cell. And now that F minus cell will become F plus, as you see right here. And then what could happen is this factor, the plasmid, could integrate within the chromosome. So we'll go ahead and look at that slide right here. We have the F plus cell, the newly acquired F factor now makes this an F plus cell. And what could happen is that the F factor could integrate within the chromosome, and then it converts this F plus cell into something that we call HFR cell, high frequency recombination. So again, the cell would go from being F minus to F plus, and then once that plasmid is integrated into the, so you have the plasmid integration into the chromosome, then that cell will become HFR, which is a high frequency of recombination. That's what the HFR stands for. HFR happens when the factor has become integrated into the chromosome. Okay, so on the last slide, we left off with the HFR cell. Once you have this HFR cell, this has that F, that fertility factor in there. So this can possibly come into contact with another F negative, a F minus cell, and what would happen is that the HFR cell will begin replicating its chromosome, as you see here. This piece that is getting sent over to the F minus cell is a part of the parental strand being sent over. So this HFR cell can transfer DNA to the F minus cell. The problem here is the chromosome that is being transferred from the HFR will break off. So I'm going to just kind of cross it off. It will break off before it's completely transferred. So not the entire HFR copy would be sent over to the recipient cell. Once within the recipient cell, the donor DNA will combine with the recipient DNA, and the recipient will remain F minus, because remember, it did not receive a complete F factor during the conjugation. So even though this HFR cell sent over some of its DNA, okay, even though that happened in this example, the recipient cell still remained F negative because the HFR cell did not get a chance to send over all of its DNA, because the chromosome would have broken off before it was completely transferred. But the recipient cell is considered a recombinant cell because it still got some of the DNA. It's shown here in like this lavender color. It still got some of the DNA from the recipient cell, but it did not get the F plasmid, so it remained F negative. The lavender color, that genetic material got sent over to the recipient cell, which makes this a recombinant cell. It's recombinant because it has its original DNA, which you see here in the dark bluish, and then it has the lavender DNA, which came from the donor cell, the HFR cell. But it did not get this yellow portion, which is the F factor. It did not get it because it broke off before all of the DNA was transferred over. Okay, so the process of conjugation is very, very useful for us because conjugation allows us to map the location of genes on a bacterial chromosome. So what we do is a researcher would allow conjugation to take place for 1 minute. And after 1 minute, the conjugation is disrupted. And what we would do is look for certain traits, certain new traits in the recipient cell. So if after 1 minute the conjugation is stopped and we see that the recipient cell now has these new traits, it's able to now make threonine and leucine, which are amino acids, then that means that those came first because those were the first genes that were sent over after only 1 minute. So if, let's say, the F minus acquired the ability to synthesize threonine, then that means that the threonine gene is located early in the chromosome between 0 and 1 minute. If after 2 minutes, the F cell now becomes threonine positive and leucine positive, meaning that now it's picked up the ability to make leucine, then that means that leucine is the second gene on the chromosome. So this is exactly how genes are able to be mapped. So once again, the conjugation is allowed to take place. We disrupt it after 1 minute and we see what the recipient now has the ability to make, for example, in this case. And whatever newly acquired trait it has first, that means that that was first on the chromosome. Just to make sure that this point is clear, let's say, for instance, we continue conjugation again for another minute and we let it go for 4 minutes, and then suddenly we have gene X. Now the recipient now has the ability to make X, product X. Then that means that X was third on the chromosome in location. And let's say we let it go for another minute, and then the gene is now able to make product Y. Then that means Y was fourth on there. First it was threonine, then it was leucine, then it was X, and then it was Y. So we're able to map these genes and figure out where they are in relative positioning with each other. Yet another mechanism of genetic transfer between bacteria is transduction. Transduction is a process where bacterial DNA is transferred from a donor cell to a recipient cell, but it happens inside of a virus that infects a bacteria called a bacteriophage. In order to understand transduction, we look at generalized transduction, where we look at the life cycle of a transducing phage of E. coli. So we're going to start right here where the phage infects the donor bacterial cell. So the phage is in there. An infection has taken place. Now what would happen is during the phage would begin to reproduce. So it would start making DNA and proteins. Of course, it's causing the host bacterial cell to make these pieces for it. As you know, the virus will basically reprogram your cell to make its parts. So this is what's happening. The bacteria is making the phage's parts for it. Now let's move over here to the third illustration. So as the bacteria is constructing the phage's parts for it, it's going to pack the phage DNA into the capsid. As we can see right here, the phage DNA is pictured in pink. So as the virus is being assembled, the phage DNA, the viral DNA is getting packed into that capsid. But sometimes the bacterial DNA gets into the capsid. Now, let's say that it packages up some bacterial DNA into the capsid, and then it bursts and it releases all of the new viral particles. What would happen is that each of these viruses can now go and infect a new host cell. So let's follow the one that got bacterial DNA packed into it. So here we have it, and once it infects a new host cell, this phage will inject the genetic material from the donor cell into this new host cell. And what would happen is that that donor bacterial DNA would be incorporated into the recipient's DNA. And now you have a recombinant cell. So let's just repeat this. Overall, I'm going to put it in green here. Okay. Overall, what happened is that you have a phage infecting a bacterial cell. Let's call this bacterial cell cell number 1. When the phage DNA is injected into this cell, the cell now is going to start making parts for the virus. That's how viruses work. Hopefully you remember how viruses work. They take over the cell and they make the cell will start making the viral particles. So here in picture number 3, when the phage is getting the DNA packed into it, it should have its own DNA packed into it as pictured by the pink, but sometimes it gets the cell bacteria number 1, it will get its DNA packed in there instead. And then when it gets released, now here's this phage with the DNA from bacteria number 1, now getting that DNA injected into a new cell. Let's call this bacteria number 2. So bacteria number 2 now has a little piece of number 1's DNA incorporated into its chromosome. And now this is considered a recombinant. Okay, so overall, we just finished covering quite a few ways in which genetic recombination can occur. In eukaryotes, we saw that it can occur with crossing over. And crossing over is when two homologous pairs of chromosomes become attached like so. Okay, so there goes a maternal chromosome and a paternal chromosome. And then some of the DNA from the paternal chromosome gets onto one of the sister chromatids of the maternal chromosome and vice versa. And now you have, these particular genes have recombined. We have different sources so that the offspring could inherit different combination of genes that were not seen in the parents before. Then we have transformation. And with transformation, we saw this was the example with Griffith's experiment where we looked at the encapsulated bacteria versus the ones that were not capsulated. So in transformation, we know that the genes are transferred from one bacterium to another. But what happened here is the fact that the newly acquired genes were picked up by the bacteria, by the living bacteria from the environment. When some bacteria die, they would lyse and their DNA would be put into the environment. And then depending on the species and the environment, Other bacteria that encounter that DNA could take up the DNA and integrate it into their own chromosomes, and then that would be a recombinant organism there. It could be a recombinant bacteria because it has genes from two different sources. The other one that we looked at was conjugation, and conjugation is different from transformation because it requires cell-to-cell contact, and it also required that the cells had to be of opposite mating type. So for this one, we looked at the F minus cells and the F plus cells. So the F plus that had the fertility factor could possibly cause a F minus cell to become F plus. Then we just ended off with transduction, and transduction involves viruses. So the new genetic material is going from one cell into another cell because of a virus. So now we have plasmids and transposons left to talk about, and both provide additional mechanisms for genetic change. They occur in both prokaryotic and eukaryotic cells. But from in our class, all we do is kind of look at the role in genetic change in prokaryotes. So hopefully you remember that plasmids are they are self-replicating circular pieces of DNA, and they contain genes. And they are much smaller than the bacterial chromosome, and we find them in bacteria. We also find them in some eukaryotic organisms, but mainly in bacteria. The F factor is one that we call a conjugative plasmid. So again, this carries the genes to make sex pili for the transfer of the plasmid to another cell. This, of course, is when one cell will come into contact with another cell. The projection of the sex pili would connect them, or they could stick together. Depends on whether they're gram-positive or gram-negative. And the genes can be transferred from one cell, from the donor cell to the recipient cell. So that's one type of plasmid, the conjugative plasmid, the one like the F factor. Now, typically, plasmids are dispensable, meaning that they're not really necessary, okay? I say that plasmids are like having a DVD player in your car. An engine is necessary, so that's not dispensable for your car to run, but a DVD player is not necessary, so that's something that's dispensable. It just gives all it does is give the recipient cell, or the cell that has the plasmid, rather, it gives the cell that has the plasmid something really cool, some really cool characteristic, like the F factor. But there are some that are what we call dissimilation plasmids. And so these are ones that are where the genes are crucial to the survival and the growth of the cell. So again, dissimilation plasmids are very crucial for the survival and the growth of the cell. These are not dispensable. So for example, it might encode enzymes for catabolism of certain compounds in order to break down certain compounds. Maybe there might be some unusual sugars or hydrocarbons that need to be broken down, and it's important for the cell to break this down. So a dissimilation plasmid may carry the genes, the catabolic genes, to break that down. Then there are other plasmids that code for antibiotic resistance, and these are R factors or resistance factors. That's what the R stands for. And these have significant medical importance because it's associated with having antibiotic resistant bacteria. And then there are other plasmids that code for endo or exotoxins. So these would enhance the pathogenicity of a bacterium. So there might be, for example, there is a strain of E. coli that causes infant diarrhea, and you might know it as traveler's diarrhea as well. And this strain of E. coli carries a plasmid that codes for toxin production and for bacterial attachment to intestinal walls. So this is, going to cause it to become more pathogenic and more virulent. And there's also other plasmids that contain genes for the synthesis of bacteriocins. And bacteriocins are proteins that kill other bacteria. It's kind of cool because it's like they wage warfare against each other. This slide is just showing a really cool picture of a plasmid. This one right here in picture A is from E. coli bacteria. And B is a diagram of an R factor, which has two parts. Now remember, the R factor carries genes that cause the host cell to be resistant to antibiotics and toxins and things like heavy metals. Okay, so again, these R factors contain two groups of genes. One group that it contains is the RTF, and that includes genes for plasmid replication and for conjugation. Then there's another group called the R determinant, and these have resistance genes. And these resistance genes, which I'll show here in the green, these resistance genes code for production of enzymes that will inactivate certain drugs and toxic substances. Of course, the R factors, they present a lot of problems when it comes to, medical issues, because it's hard to treat these diseases with antibiotics, these infectious diseases, because they are resistant to so many drugs. And the accumulation of the resistance genes, of course, here in the green, I only showed 4, but there have been genetic maps of resistance plasmids that carried so many resistant genes. We showed like there have been resistant genes for sulfonamides, for streptomycin, chloramphenicol, tetracycline, even genes that are resistant to mercury. So it's really difficult to treat the bacteria with all different types of resistance. All right, so moving on, the last thing we have to talk about will be transposons. And transposons are small segments of DNA that can move from one region of a molecule to another. And we first discovered this in corn. The segments can move from one site to another site on the same chromosome, or they can move from one chromosome to another. Now, this can be dangerous because if a transposon gets inserted into a gene and inactivates the gene, then we can have a problem. But fortunately, this is a very rare occurrence. All transposons contain the information for their own transposition. So there are, for example, insertion transposons. And this will contain a gene that codes for an enzyme called transposase, which can cut and reseal DNA. We also have complex transposons, and these carry other genes that are not connected to the transposition process. Like some may contain genes for enterotoxin or antibiotic resistance. So I'm going to end this particular discussion on Chapter 8 on this slide. Please make sure that you look over it over and over again. And of course, if you have any questions, I am here for you. Send an email. I'll stop by my office to discuss any of this. I'd be happy to talk about it with you. I hope that it was pretty clear. I tried to keep it as simple as possible, and in terms of it just being really, really straightforward. So enjoy. Happy studying.